Does anybody need a stapler for their homework?
Do you want that sheet you gave us with it, or just our?
I need things numbered. That's all I care about. So no, you don't need to have that as a cover sheet or anything like that. I just need things numbered, and we'll sort that out as we go forward. Okay, that's fine. It's numbered, so there you go. So you have
One on it?
what?
What number do you want to put on top of it? One, assignment one?
You could do that, assignment one, but I will have them together. I want the numbers of the questions going down. That's the important, that's the important thing, because I do them all at once and I line up the questions and 1, 1, 1, 1, 2, 2, 2, 2.
Okay.
So. That's the challenging part. So. Work on passing those assignments forward. Is anybody here today from an attendance perspective that is here but not turning in an assignment? So if I get an assignment from you, you're here, right? And we got a couple of students that are excused. Yeah.
So I was going to print mine off, it's all done, but then I kind of got stuck in Rome on one of Marco's field trips.
Okay.
And I printed off right after and bring it to you.
Yep. I should be in the Marshall here for probably a half hour, 45 minutes, and then I gotta get to the dairy. I won't be there in a second. Anybody else have any concerns about what they're turning in today? So anybody else got stuff to turn in? Okay. So we're dropping fast here. Does anybody know why that might be? Am I that bad? Is it weather? Or just too many field trips today? Or about half the registered number of students here? Does anybody know anything that I don't know? Okay, so getting started. Well, those of you that want it, there'll be a review session at the end here. We'll do our 5-minute break. We got the books on reserve. Written assignment 1 is due now. Written assignment 2 will due a week from today. There's a Word file of that up on Blackboard if you want to use that, or if you want to type something separate. The class has said hard copy so far, so we'll see how that holds up as the semester wears on. So, quick review. Vocabulary pairs. What pairs with superior? Inferior. Caudal. Cranial. Anterior. Posterior. Ventral. Dorsal. Lateral. Medial. Proximal. Distal. So close, or if you're doing, I don't know, did you guys watch Sesame Street growing up?
What is that?
A little bit. Grover, near, far. Proximal, distal. Superficial. What pairs with that? Deep. Flexion, extension, abduction with adduction. So last time we talked about carbon and its versatility. It's the basis of all life. That carbon atom's ability to form four different bonds gives it a lot of flexibility, allows it to do a lot of different things. If we just add hydrogens to a string of carbons, we create a hydrocarbon. Very simple, very basic organic molecule. The thing about it is it's non-polar. It tends to be very unreactive. So if we think of our petroleum products, and we think about oil for lubrication or grease for joints, we want them to be non-reactive. We don't want them corroding the metal, and they do that very, very well. If we add water to the system, we get into trouble. So we ended up with a discussion of functional groups, the things that are added to these base hydrocarbons that are going to give them their individual character. So why do we care? Depending on which functional group's there, that's going to determine what we're dealing with, or what organic compound class we're dealing with. So this becomes important when we're thinking about polymers. Has anybody ever heard the word polymer before? Some of you have, some of you haven't. Polymer, poly, many. So we're coming from the Greek, many parts. And our cells manufacture polymers, strings of repeating units that become very, very large molecules. And when we do that, we connect those together just like we would the cars in a train, and eventually we get that train that's got a distinct character to it. So individual train cars as individual molecules that we will call monomers. So a string of monomers is what makes up a polymer. And we deal with a lot of different polymers in biology. We'll also deal with that. Most of you are wearing something synthetic. Those are polymers of different molecules to create those plastic threads. Small repeating unit. So cells manufacture polymers by hooking, putting those monomers together. And those hooks are when we have interactions with functional groups. The reactive parts of any molecule are the functional groups. They're going to form the linkage, they're going to form the hook. So we'll have a string of monomers, some sort of repeating unit over and over and over again. We put those units together, we'll call them a polymer. As I said, we'll think about the links in the train. In our train systems, these connections here, these knuckles that hook the cars together are standardized. Every car has one at either end. Doesn't matter what's in between, what's built, what the superstructure is, whether it's a tanker, whether it's a box car, whether it's a cattle car, whether it's a flat car. They all have that base down below that has that connection at either end. And then they'll form that linkage in a consistent and definite manner. So how do they join together in biology? What are we doing here? We're going to involve the addition or subtraction of a water molecule. We'll take two monomers, we'll bring them together. We pull out a water molecule, those two monomers are hooked together. We reverse that process, we force a water molecule in, we break that bond, we separate those two monomers once again, and you should have a handout related to this. So two monomers, at somewhere on those monomers, you're going to have a hydrogen site, and then somewhere on that monomer, you're going to have a hydroxyl site. They can be a lot of different things, but ultimately you're focusing on this connection here, this H2O. So you should have this sheet. I think I gave it to you. It's not in color, but I think it's in full size. So if we're making a chain, we have a chain of monomers, a short polymer. We're going to add one more unit to it. Each monomer has a hydroxyl side and a hydrogen side to it. We pull that water out, and we form a bond between that third and fourth monomer to make a longer polymer. This is called a dehydration reaction. Does anybody know why that might be? You're getting rid of the water, you're pulling water out. So the removal of water to make that chain just that one link longer is called the dehydration reaction. If we take that water molecule and put it back in, we force that back in, we're going to break up that third and fourth monomer, make that polymer just a little bit shorter. And the water parts, hydrogen goes on one side of the gap, a hydroxyl group goes on the other side of the gap. This is called a hydrolysis reaction. Hydro meaning water, then molysis means break, rip apart. So we're ripping apart with water. Hydrolysis reaction or hydrolysis might be an easier way to remember that. Most people pronounce it hydrolysis. So we have a number of different monomer sets that make a number of different polymers, but they all connect with this dehydration and hydrolysis reactions. So for creating polymers, if you're a plant, you can take a series of glucoses, join them with the dehydration reaction and get the polymer starch. So that only happens in plants. Same thing with the glucose, slightly different linkage, but same reaction. Dehydration reaction, we can create cellulose. Same building block, slightly different configuration. That different configuration gives us cellulose, that plant cell wall. Another type of monomer that we'll find in the body are amino acids. To make proteins, we string together a combination of 20 different amino acids. And this could go on for thousands and thousands of amino acids, and we eventually make that protein. Nucleic acid is the building block of DNA. We put a string of those together, we get the DNA molecule or we get the RNA molecule, depending what the building blocks are. But again, dehydration reaction connects those monomers together. So making a different type of monomer. This is where we come back to what we started the lecture with. We have a base hydrocarbon, some sort of hydrogen-carbon combination shape. Then we add functional groups to that hydrocarbon, replacing carbon or replacing hydrogens. Well, maybe a carbon or two. But the addition of those functional groups defines which monomer class it ends up in. So if it's an amino acid, it's going to have a carboxyl, and it's going to have an amino group. If it's a sugar, it's going to have the alcohol group. Yes?
Can you put this up for me real quick? Monomers are single, and then a bunch of them combined together makes a polymer?
Yes.
Okay.
So a string of monomers equals a polymer. So we're making those basic building blocks here. We start out with some sort of basic hydrocarbon, and we'll show that as we go through. We add a few things to it. The functional groups give it definition. What group does it belong to? And the functional groups will also give it its reactivity. The functional groups are where those dehydration and hydrolysis reactions occur. So if we're looking at our monomers that we're going to deal with in this class, we have glucose, amino acids, nucleic acids, and fatty acids. So if we're going to build those, what components do we find, or what functional groups do we find in each of those? This table didn't come out quite as clear as it might have, but you do have this in the handout today. So the transition from color to black and white was pretty limiting. So all of those monomers have a base or a backbone that's a hydrocarbon. We've already discussed that. And then depending on which functional group, the four we discussed, hydroxyl, carbonyl, carboxyl, or amino, will determine which of those classes we are in. So glucose, you're going to have mostly alcohol groups, the OH, the hydroxyl group. And then at the end, you're going to have a carboxyl group that allows that linear molecule to become a ring if it needs to. Amino acids is going to have all of those, depending on which amino acids specifically we're talking about. But the big two are this carboxyl group, which is at one end, and the amino group, which is at the other. Every amino acid has a carboxyl at one end and an amino group at the other. By definition, what separates the amino acids and therefore the protein away from everything else is that amino group, the presence of nitrogen. The N up here denotes the presence of nitrogen. We'll talk more in depth about that. A nucleic acid is made up of 3 different parts. The sugars have exactly what you'd expect. We have the match here and the match there. The sugar, and there's a phosphate group, and then there's a nucleic nitrogenous base. Nitrogen implies that amino group, and yes, in fact, that is there. Fatty acid has that long chain of hydrocarbons that has a carboxyl group down at the end. By definition, by naming, it's put into that category because that carboxyl group is there. So the linking that occurs in these quote-unquote monomers to make them polymers occurs where the the hydrogen ions and the hydroxyl ions are. And this is where the reactivity occurs. So where are things hooked together? Where do things connect? There are groups there, but the ones that are circled contain the ones that are going to form during that dehydration reaction. There can be other reactions, cross reactions, but they're not going to be the ones that are going to create that polymer from those monomers. So you've got the backbone, hydrocarbon, chain of carbons that runs through the molecule, and the hands or the hitch are going to come from those functional groups, where we connect one monomer to the next. Any other questions about polymers and monomers? Is that making sense so far? Okay, how do we use that? We're going to move on to carbohydrates. So carbohydrates, the name implies carbohydrates. Base chemical formula is a ratio of 1 carbon to 2 hydrogens, H2, and 1 oxygen. So if we go back to our chart from a little while ago, we're adding oxygen to the mix. We start out with our hydrocarbons. What changes moving to carbohydrates is the addition of oxygen. And from an atom count, you're looking at about 1/4 of the atoms involved are oxygens. Everybody find that page that wants to. So for carbohydrates, generic chemical formula, we have our N's down here to denote that there's a ratio of 1, just 1 N, 2, 2 N's, to 1, just an N. So 1 to 2 to 1 roughly. It doesn't always work out perfectly, but that's about where it all fits together. So carbohydrates or organic compounds, and that's because of the presence of carbon in them. There's a range of different monomers. They can either be floating around singly like glucose does. Glucose is the energy currency in the body. moving from organ to organ, floating in the blood, very tightly controlled. We get into diabetic issues and hypoglycemia issues. The single sugars can also, depending on the textbook you're looking in, be called monosaccharides. This is more old nomenclature on some level. When we're looking at these single sugar units, these single monomers, they can be 3 carbons long in a chain and up to 7 carbons long. So there's a variety in biology that will occur depending on what their function is, depending on what their purpose is. So the backbone of these monomers is 3 to 7 carbons long, the chain of carbons that runs through it, 7 to 3 carbon atoms. 6 carbon sugars are the most common, and we're usually looking at glucose. But there are about a dozen different isomers with the same number of carbons, same number of hydrogens, same number of oxygens, 6 to 12 to 6 that can occur, and those slight differences give them slightly different character. The second most common rings that you're going to find are those with 5 carbons in them. We can do those single building blocks, those single monomers, to huge polysaccharides, huge strings of monomers, thousands and thousands of monomers long. So polysaccharides are chain of monosaccharides. Generically speaking, polymer versus the monomers. So again, in plants and animals, on this planet anyway, you're looking at glucose. Glucose is the dominant molecule, organic molecule on this planet. Basis of most of life, there are certain things that will derive their energy from other compounds, but most, we need photosynthesis to create glucose. And that glucose is passed around the environment, passed around the world to create things to support life. These polysaccharides can be linear in fashion, just a long string, or we may have something that branches at certain points. Depending on the purpose, depending on the use, we may have branching linkages during those hydration reactions. We may have linear chains during those reactions. So between that single unit and those huge strings, we have a lot of different variation. So carbohydrates, the 2 most commonly that we'll find in cows are the pentoses. You're going to find a lot of those in DNA and RNA. The ribose function is there. The hexoses are going to be usually in the form of glucose, except for its isomer galactose, which we'll find in the udder. That glucose is going to be all over the body, 6 carbons long. Anytime you have an OSC as a suffix, you're looking at a carbohydrate, usually a specific sugar. So lactose, sucrose, fructose, hexose, monos. So given the range of carbons, 3 to 7 in the chain, there are a number of isomers within each number. Does everybody here know what an isomer is? We alluded to those with the butane. You can have a branched form of butane. You can have a string form of butane. Same number of atoms in a different configuration. We can take a pile of Legos. We can put it together a number of different ways. Those different ways are called isomers. So here's an example of what we're dealing with a 6-carbon backbone. So the nomenclature or the standard, what would you call that? Convention. When you're dealing with organic compounds, anywhere you have an intersection of two lines, there's a carbon atom implied in that location. So if we're looking at our glucose here, we have 1, that C up here, and then 1, 2, 3, 4 at those drawn intersections, and then our 6th one down here at the end. So depending on where those hydroxyl groups are, which side they're on, will determine which isomer is present. All these have the same number of carbons, the same number of hydrogens, the same number of oxygens. Glucose being the most prevalent, the most common appearing. So if we're looking at our diagrams in chemistry, we can either draw everything out with a letter representing an atom at each location, or to clean things up, make things faster to draw, we do that assumption that in each of those intersections, there is a carbon atom sitting there. Especially on larger molecules, this shorthand allows you to move a whole lot faster. And then when we notice the glucose can occur either as a ring form or a linear form. It can fold back on itself. We've got an OH group here, a hydrogen here. It's going to fold around, and that's going to give us our dehydration reaction. So with glucose, you're going to have 1, 2, 3, 4, 5 carbons in the ring. That hexose forms a 6-sided ring, but one of the spaces is an oxygen, and then you've got 1 carbon hanging off of that ring that can react with the next molecule. So glucose is the main one you're going to find in the body. Why is glucose important? Reason number 1, glucose is the main fuel molecule passed around the body to accomplish work. So the energy to do things. So it's the fuel that gets things done, the gas that makes the motor run, the energy that makes the nervous tissue run. Nervous tissue prefers glucose. And as we make a change from glucose to not glucose, if we're on a prolonged hunger strike or something like that, we change to keto acids. There's a period of fuzziness where your brain isn't working quite as well. As it changes over from its preferred source of energy. We can also use fat for energy, but that's kind of stuff we put into storage and then take it out later. The second reason glucose is important, it becomes a base building block for pretty much everything in the body. We can use glucose or a variety of other monosaccharides or monomers to furnish or donate a carbon skeleton to make something else. Manufacture other organic molecules. We can use it to make fat molecules. We can use it to make protein, amino acids. We can use it to make pretty much anything you're going to find in the body, connective tissue, all eventually leads back to glucose. The liver can take glucose, for example, and make amino acids, taking those carbon molecules, that solid, breaking it up, chopping it up, rearranging it, gluing it back together, and we've got an amino acid. So those of you that took nutrition, what are the step one, reason one and reason two? What are those? How did we summarize them last fall? Burn and build, right? We can take that glucose and we can burn it for energy, or we can chop it up and use it to build things. What we're going to add in this class is reason number 3, that glucose can be stored for later use. So we can store glucose as glycogen. We take the monomer glucose, we put the string together, and we form the polymer called glycogen. We can form glycogen in the liver. We can form glycogen in the muscles. Who here has done carb loading? Big pasta dinner the night before a game, trying to get your muscles carb loaded up. What you're doing is making glycogen in your muscles for use during that physical activity. Little extra pasta, lots of glucose absorbed. That glucose goes to your muscles to be stored in glycogen for the big event. So in addition to burn, use it for energy, we use it for structure, we're going to build with it, or we're going to store it. We're going to save it for later. We can either burn or build later. So it would eventually all be burned up, probably. But burn, build, or store with our glucose molecules. So burn, build, or store to help remember those 3 purposes. So monomers, we talk about glucose as being dominant. We'll have some disaccharides that are pretty important in the body, at least in biology. Sucrose, table sugar, is 1 glucose and 1 fructose stuck together. There's a dehydration reaction that occurs here to form this bond. Lactose, we've got glucose, galactose. We're forming that dehydration reaction there to form that bond. Maltose usually occurs when we're breaking starch down in the gut. The starch is broken into 2 units, cutting off 2 at a time, and then a different enzyme comes along and hydrolyzes between the 2 glucoses. The starch, a string of glucose, broken down 2 units at a time, we'll have maltose in the gut. That'll be broken down into 2 glucoses that are absorbed. So we're thinking about our sugars, our monomers. We can go from 2 to thousands in a string or a branched sequence. So the polymers we're going to talk about for glucose, we mentioned 2 already in this class. Well, we mentioned them all. We're putting them all here together. Polymers that can be made from glucose. We have a starch that's made in plants, and that's the energy storage form of glucose in plants. So potatoes, corn, wheat, all are saving that starch for the next generation. Put that with the embryo, and that becomes the energy for the next generation of plants to get their start. Glycogen is the storage form of glucose in animals. Same string, slightly different configuration, but we store our glucoses together. Till we need them later on, we can break them apart. We can break down that liver glycogen, we can break down that muscle glycogen and use the individual glucose units for whatever is needed locally. Cellulose, plant origin. It is a string of the monomer glucose. This is a structural carbohydrate. Once it's formed by the plant, that stuff cannot come back. Makes a commitment to that cell wall, it stays in that cell wall. Plant dies, then the fungus and bacteria in the soil can break it apart, but once plant makes that, it's committed to that. If we take a look, I think you have this picture in your set of this diagram here, cartoon versions of starch in the potato, glycogen in the arm of the producer here, and then cell wall in these plants that are grown here. We're not going to spend time in this class talking about what is this, is an alpha 1,4 linkage or a beta 1,6. I don't care about that for this class. But looking at those cartoons, those are fairly representative of the shapes that occur in starch, in glycogen, and cellulose. So just looking at them from a visual perspective, what do you see as differences between those cartoons? Those 3 different polymers made of the same monomer connected in a slightly different way. What do you see there as differences? So turn to a neighbor and see what differences we can come up with. What do you see? So the idea behind this diagram is that it's fading back into the screen, and then we have these individual pink hexagons are individual glucose units. So what do you see there as differences? Anything? Yes. Okay. That's a good way of putting it.
Starch is just kind of, where it's all balled up, a lot of small parts.
Okay. So we have what cellulose forms with these three strands is sheets. They're laid out flat. They're cross-linking between these chains. These chains tend to be fairly linear. And how do you think we get that, given what you see in the diagram? See those hexagons flip-flopping? So we're taking out the kinks and we're developing something that's pretty straight. Oxygen is up, oxygen down, oxygen up, oxygen down. In these, the oxygen is always up. And there's a spiral or a coil to it. So formed here, you're also with glycogen going to get some branching. So from one single junction point, you might have a couple different strands going out. So given their function or given what you're doing with them, you're trying to create sheets here that are going to hold the plant up, that are going to keep that plant cell from exploding when it's exposed to water. Here, you're trying to create a molecule that can be taken apart easily again. And we'd like it to be done quickly in glycogen, and that's what the benefit of branching is. Instead of just working on one end, if I have a whole bunch of ends, I can release a whole bunch of glucose very quickly. So after I've carb loaded, a long time ago for me, during that activity, I can release glucoses a whole lot faster if it's branched. Don't need that speed in starch in this diagram, but there is a branched form of starch as well moving through. So depending on what their purpose is, that's how they're linked differently together. Each of those bonds, each of those dehydration reactions is slightly different. So writing that out, polymers made of glucose. Starch is for storage in plants. Glycogen is for storage in animals. Cellulose is meant to build plants, and those glucose molecules are not retrievable once again. So when we're thinking about the grass that's dead and left behind, it can, if it's a perennial, it can pull out pretty much everything to recycle at a later time, except the cellulose. So that's all that's usually left in the stalk. At the end of the life cycle, that's all that's left in the leaves. At the end of the life cycle, or mostly what's left in the leaves that fall on the ground, just cellulose. So why don't you take a break? We'll come back and we'll test your understanding of osmolality. Make sure we've got that concept down. So we'll start again at 7 of. Thank you.
Before you both express an interest in marriage.
So what we need from you is that you have to have completed 30 credits. Have you done that yet? Not yet. So we'll focus on next fall. Getting going, you have to have completed a year. I don't know if you transferred anything in or not. And we'll focus on next fall. Okay, let's come back. So get out your letters and we'll test our understanding of osmosis and osmolality. So if an animal cell is in a hypertonic solution or a hyperosmotic solution, which of the following is true? The solute concentration is higher outside the cell and the water concentration is higher outside the cell. Solute is concentration outside the cell is lower. Water is higher inside the cell. C, solute concentration is higher outside the cell and water concentration is lower inside the cell. Solute concentration is lower on the outside and water concentration is lower on the outside of the cell. So which of those is likely right? So hypertonic by definition tells us what? More solute. So the solute concentration outside is higher. That limits us to A and C. If we have more solute in that outside, we're going to have a lower water concentration outside the cell compared to the inside of the cell. So if we're doing the same thing with hypotonic or hypoosmotic, what's the ultimate hypotonic, hypoosmotic solution? Anybody remember? Water. So we know that the solute concentration is lower outside the cell and the water concentration is higher outside the cell. So we're looking for answer B. So with the illustration of the exploding and shrinking cells that we had, what's happening? Is water moving? Yes, it is. Is solute moving? Not initially. If there's a rupture, it will move. So water is not moving. Water is moving. Solute is not moving. That gives us answer C. So any questions on this? So if you can figure out where the concentrations are highest, remember things move from high concentration to low concentration. You can figure out what can move and what can't move. You should be able to figure which of these scenarios will occur. So we've talked about water. We spent some time with carbohydrates. We're going to move on to proteins. So proteins is a polymer of amino acids. So we take a string of amino acids, we put them together, and we make what's called protein. And we're usually looking at hundreds, if not thousands, of amino acids in a specific sequence. Those of you who had nutrition, we're talking about 20 different amino acids put together in a particular order. So if we look at which of the big four are present, all of them are present. So you've got oxygen, you've got hydrogen, you've got carbon, and then by definition, you have nitrogen. Nitrogen is the defining element of protein. That amino group being present determines that it is protein. So this is a review for some of you who've had nutrition. For those of you that haven't had it, we talk about the addition of the amino group gives protein its unique character. And most of our measurements of protein involve measuring the nitrogen and then multiplying by a factor to give us the total protein. Usually we use 6.25, but it depends on the protein or types of protein. Most animal feeds are 16% nitrogen. So if I want to get the total amount of protein, I multiply the amount of nitrogen by 6.25. 16 goes into 100, 6.25 times. And that's where we get that correction factor for that average animal feed protein. So polymers are formed when amino acids come together. The amino acids are the monomers, and I will have a tendency to abbreviate or show amino acids as that double A or the double A and the S if I'm really getting specific about there needs to be multiple ones involved. So that's sort of convention. I don't think it's official, but that's the way most people do it. Protein is made up of combinations of 20 different amino acids, more or less found in nature. We think about 10 essential and 10 non-essential amino acids. There may be some other variations depending on what animal we're talking about or what environment we're in, but we're going to focus on 20 for our dairy cows. So 10 essential amino acids and 10 non-essential amino acids. We won't focus too much on the difference in this class of what that means. We've got 20 building blocks that we're got to choose from. If you're into Legos, you got a tray, you've got 20 different options to pull out. Depending on which ones you pull out, will determine what you can create. All 20 follow the same basic blueprint. They have a constant region and a variable region. The constant region is a chain that runs through each amino acid. We have an amino end. We have a middle carbon. And then we have a carboxyl at the other end. So amino acids, you got the amino at this end, you've got the carboxyl acid at the other end. All proteins are going to follow a chain that repeats NCC, 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 NCC. What's different among the 20 amino acids is what hangs off the middle carbon. And that variability is denoted with an R. R can be a wide range of things, and we'll give you some pictures of those in a minute. But this part is the same for every amino acid. This part is different. That's how we tell one amino acid from the other. What is the R group? And that will tell you which amino acid we're dealing with. So a generic amino acid, something similar to what we got on the board there. That R is the unknown. That R is the difference part. So you've got R group can be something as simple as a single hydrogen ion. Glycine has only that hydrogen ion sticking off that middle carbon. To very complex hydrocarbon rings with many attachments. Oh, we've got that common part for all our amino acids. And we've got the different part. The different part determines which of the 20 it is. So, and we're dealing with our cows proteins. We've got 20 different R groups. The backbone of a protein is going to be that repeated pattern. Every protein at its heart will have this same repeating backbone where the red line is a what? A bond formed by what? What?
Would that be the carboxyl?
The carboxyl end is the C end. And you've got a carboxyl end here and a nitrogen here, amino group. So this is what? Formed by a what?
Water.
Removal of a water molecule to get the monomers going in order, whatever that may be. So dehydration reaction forms that red bond we're illustrating there. Given this common backbone, you've got this long string of atoms together, we can twist around that molecule. And depending on how the molecule twists, we'll give it the final protein its shape. So here's oxytocin. We have blue, 3-letter codes here. Each of these represents a specific amino acid by name. So you have glycine right here, NCC. You've got leucine, NCC. Leucine's got this chain hanging off it. NCC, you got proline with that ring hanging off. But if you work through that oxytocin, got NCC, NCC, NCC. We can work through that backbone that trails from one end of the molecule to the other. What the difference is what's hanging off that middle carbon. You see glycine, not much of anything hanging off there. We've got tyrosine's got a big ring associated with it. If we're dealing with our amino acid, here's our generic amino acid. We've added the hydrogens in, that hydroxyl group, fully illustrating the carboxyl group down there at the end. We take 2 of those together, the carboxyl and the amino group. Dehydration reaction forms what's called a peptide bond, the bond between the nitrogen and the carbon, formed with a dehydration synthesis reaction or dehydration reaction. Short strings of amino acids are called peptides. So in this illustration, we have a dipeptide, 2 amino acids stuck together. Those of you that are interested in nutrition, there does seem to be a requirement by the microbes for peptides. Short chains of amino acids, you seem to get higher microbial yields when those are present. So we talked about that. So close up on that. You've got that peptide bond and the backbone starting to form there that puts all the way through our protein. So this diagram, I think I turned it sideways for you, is talking about all the 20 different options that we might occur in our non-essential and essential amino acids. So we have glycine up here as one. And then we've got them grouped by their reactivity with water. We have hydrophobic R groups, alanine, valine, phenylalanine, proline, leucine, isoleucine. All are hydrophobic in their interaction. So if we look at our color coding up here, we've got hydrogen is our open little circles. On mine, the solid black ones are carbon. So what we see here is mostly just hydrogens and carbons. So we have R groups that do not like water. They're going to do whatever they can to steer the molecule away. So the R group almost becomes like a rudder, a steering mechanism. Hydrophilic. All of these through here on my diagram, what do you notice is a difference between this group and this group? What? I can't hear. Just looking at it from a drawing perspective, what do we see? We got more color down here, don't we? So we've got red, which is oxygen. There. We've got yellow, which is sulfur. And we've got a couple of blues, amino groups. Almost all of these have a charge associated with them because they have something in addition to hydrogens and carbons. So if they've got a charge to them, they're going to like being around water. So they're going to pull the molecule toward water, make it water soluble. So hydrophilic. Then we've got 1, 2, 3, 4 down here that are amphiphilic. So what does that mean? They really haven't made up their mind yet, right? An amphibian or salamanders live on land, live in water. They can go either way on that. But the reactivity, especially focusing on the hydrophobic group and the hydrophilic group, determines the shape of the molecule when we're done. And we'll show you that over the end of this class and the beginning of the next one. So, a little closer look, a little bit more color in these. They tend to react more with water. So if we're thinking about our proteins, we can divide them into kind of flat or linear structure or round balls. So the round balls are good for blood proteins. They move easier than long stringy things. Little round go through the plumbing a little bit better than the stringy stuff. Long, thin structural proteins are great for making tendons and ligaments and bone, things that you're going to hold together over time. So balls versus long strings. Protein that contained glycine molecules, that was the one at the top of your chart there, where the R group was just a single hydrogen. That's going to be easier to spin around, to twist, than if it's got an R group that's got this big gangly thing there. That's not going to spin as nearly as easy as a glycine will. So wherever there's a glycine, we can twist, coil, and fold more easily than R groups that are much larger. Probably has to do with more of a volume filling thing, but also if things are bigger, it's harder for you to spin them faster. Just angular momentum. So our backbone, we're trying to spin, and in that spin, we create a final shape. Now, if we were probably being honest about this string here would kind of go up and down. There's an angle between those. So that way you've got something twisting around on an angle. If I put it like that, I can start alternating, making things linear. If I keep twisting, I can get a curve if I need to. So depending on how big those R groups are, which one we had in that diagram, will determine how easily things spin around that backbone or central axis. So, and then which way it moves depends on whether it likes water or not. What did we say is the solution in the body? Water. So the R groups that like water are going to move toward the water. The ones that don't are going to try to ball together, just like oil in your salad dressing. It tries to ball together. I don't like water. I want to be over here with my friends. So if we think about our group here and we designate the blue as hydrophilic and the pink as hydrophobic, how might this molecule move? So first you're going to basically segregate on different sides of the molecule. The blue is going to go to one side, pink are going to go to the other, the hydrophobic, and then the hydrophobic, since they're scared, are going to try to move toward each other. Try to form one unit where all of them are similar. Try to exclude the water. So given this linear chain that was produced, you're going to start to get a shape of a molecule based on whether the R group likes water or not. Everybody following that? So depending on which amino acid you pick, that particular spot in the protein is going to twist a certain way. So we need a set sequence of amino acids to get a specific shape. The same protein will twist the same way just based on whether the R group likes water or not. It will create the same three-dimensional shape no matter what. And that has some real benefits for life. So depending on the protein, we might say, has anybody seen Pandora jewelry? Depending on what you create, depends on what you might pick. So different bracelets, depending on what you pick or what you like. Same with proteins. We've got 20 different choices, and they can be arranged in ridiculously infinite number of combinations and sequences. So we're thinking about protein structure. The thing that ultimately determines the shape of the protein is its reaction with water, specifically the R group. The R group is set by which amino acid it is. So the primary structure, we put a certain string of amino acids together. That linear sequence is called the primary structure of the molecule. Got 20 different amino acids. They have each amino acid that has a full name, has a 3-letter code, or has a 1-letter code. Those of you that are trying to fold proteins in space with artificial intelligence and 3D VR goggles and all that stuff. You put a sequence together and then you watch how it folds together, creating new proteins, potentially can give different functions. So primary structure, the sequence of amino acids in this diagram, we have the 3-letter codes being used. The first thing that happens, areas that are meant to coil will start to coil around themselves. Areas that are meant to form sheets, which are usually heavy in glycine, will form sheets. So certain sections of the entire chain will organize. Then those sections start to interact and form our final structure of the molecule. And that molecule potentially can interact with other molecules. So primary sequence, that spit out of a ribosome. Does everybody know what I mean? You know what have you heard ribosome before? Has anybody not heard of a ribosome? We got one. So that's basically the machine that makes the sequence of proteins. It spits out that tape. That tape is in water. It starts to fold as the R groups react. That's going to form that secondary structure, which is the polypeptide chains. Sections begin to coil. Sections begin to fold. That's our secondary structure. And I'm out of time, so I will end there. And we'll pick up with that next time. Any questions? Anybody interested in a review session for this today? Take a break, 5-10 minutes, 3 or 5 minutes, and come back. If you have questions, things you want me to go over again, I will stay here until your questions are done.